Hi, I'm Mikang Sim from Kongun University in South Korea. My poster title is Reference Genome Sequence-Based Read Clustering. Let's get started. Clustering is unsupervised grouping of data based on common features. Various clustering algorithms, including hierarchical, partitioning, and graph-based algorithms, have been developed. They have been applied to an extensive range of data analysis, including sequencing data analysis, where the next generation sequencing reads from genomes or transcriptomes are fundamental materials. In this field, read clustering is the test to group the NGS read by considering characteristics of read, such as sequence-level similarity. Several clustering algorithms for sequences have been developed based on various sequence features, such sequence-level similarity and the distribution of subsequences of length k, called k-mer. Recent advances in sequencing technologies and assembly algorithms have enabled the accumulation of high-quality genome assemblies of many species, which are very useful resources for providing information of read proximity that can be used for read clustering. Here, we present a new NGS read clustering algorithm by utilizing references and their phylogenetic relationships. Our clustering algorithms consist of 4 steps. In the first step, using a set of genome assemblies of reference species as input, pairwise whole genome sequence alignment between one of the reference species chosen as a leading reference and each of the other ones. Pairwise whole genome sequence alignments are then used to create syntonic regions among the used reference species. In the second step, reads are mapped to the genome sequence of the leading reference, and physical coverage in the syntonic regions are calculated. The physical coverage information is used to further refine the coordinates of syntonic regions. The reads that are mapped to the same syntonic regions are grouped into the same cluster. In the third step, all mapped reads in the previous step are clustered by inferring the distances of read pairs in non-syntonic region in the genomes of multiple reference species. This clustering step consists of 3 substeps. First, the distance of a pair of 2 reads is calculated with their mapped position in all references. Second, only reads which are mapped to all reference genomes are extracted, and then the distance matrix containing distances of all read pairs is generated for each reference genome. Finally, distance of a read pair in the target species is calculated by the weighted sum of distances in references. The weight of each reference is calculated from the divergence time in a phylogenetic tree among the target and reference species. In the last step, the generated read clusters in the previous 2 steps are merged by using the parent read information. Specifically, 2 clusters linked by at least a certain number of parent reads are merged. In the following evaluation, we used 5 as the value of the minimum number of links. To assess the performance of our clustering algorithm, we generated simulation datasets of yeast and human genome sequences. We used whole chromosomes of the yeast genome and randomly selected 3 chromosomes of the human genome. For the yeast dataset, 2 Saccharomyces genomes were used as reference genomes. For the human dataset, the gorilla and orangutan chromosomes were used. In this evaluation, we assessed the purity of clusters, calculating the entropy for each of the generated read clusters. For the yeast dataset, most of the generated clusters show very low entropy. Specifically, all of the clusters from the distance-based clustering in the yeast dataset were completely pure. For the human dataset, we could obtain more than 90% clusters with low entropy. Finally, we tested the applicability of our clustering algorithm for de novo genome assembly. Using the clustered read sequences of the yeast and the human datasets mentioned before, the space assembler was used to make context sequences. The generated assemblies were then compared with assemblies generated without utilizing the cluster information by using the quest program. In the case of yeast dataset, there was more than 70% decrease of the number of misassemblies. And in the case of human dataset, there was more than threefold increase of N50 and more than 60% decrease of the number of misassemblies. We present a new read clustering algorithm based on syntax regions and infer the read distance in the genome sequences of closely related reference species. We believe that our clustering algorithm will keep increasing as more high-quality genome assemblies of many species are accumulated. Thank you for listening.